Hello, I'm Frances Reid, Programme Director for the World Ovarian Cancer Coalition. And throughout our event today, we're hearing from some very diverse and interesting projects around the world that fit closely with our Global Ovarian Cancer Charter goal of not missing out on opportunities to act on family history. And my guests in this session are part of a groundbreaking study in Australia called Traceback, Preventing Women's Cancers. It's a collaboration between the Peter MacMallum Cancer Centre, our Charter Champions, Ovarian Cancer Australia and others, and is supported by government funding. With genetic testing for women with ovarian cancer in Australia and their families now commonplace and enshrined in guidance, this study focuses on those who missed out on the possibilities of testing in the years as knowledge and guidance developed. I'm delighted to be joined by two women closely involved in the project. Both work for the Peter MacCallum Cancer Centre. Rachel Delahunty is a medical oncologist with an interest in gynecological cancers, working in the Mercy Hospital, and Dr. Catherine Alsop from the Cancer Centre, whose work originally led to this study. Rachel, you were one of the people who got this project off the ground. How did you find out that there was an opportunity to find these missed women?
Yeah, thank you. So there was obviously an important paper from our lab in 2013. It was led by Kath that was really important for recognising ovarian cancer as a sentinel cancer for women that carry a germline mutation in BRCA1 and 2. And that was very important for changing guidelines for genetic testing in Australia. And we have seen that has worked very well, and our testing rates have increased over time and now approach 90% at most centres. It was really a recognition of this moral dilemma that we had, knowing that there was a lot of untested women that have died, which really meant that there's families out there in the community that are untested. And it's been this feeling that people, senior clinicians, have felt really uncomfortable about knowing that they're out there and that actually nothing is being done about them. And recognising that it is an ethical dilemma, and knowing how best to approach this particular group, given that they're deceased, but also balancing that against the potential harm in not doing something about it, and the harm that could be done to those families. So it was really some discussions originally between Michael Freelander and David Botel, and then Goli Samimi, that triggered a really important workshop led by the NCI. And from that, there was essentially a framework developed, which was published in the Journal of Clinical Oncology. And then I came on board as a PhD student. It's really been my job to turn that into a working project.
An amazing project it is too. And it is a feat of collaboration, really, isn't it, Kath?
Oh, absolutely. The Australian Traceback study, it's a really collaborative project. It's spearheaded by experienced ovarian cancer researchers, including Professor David Botel, as Rachel mentioned, but it also heavily involves cancer genetic specialists, medical oncologists, including Professor Michael Freelander, ovarian cancer cohort studies that run in Australia. And most importantly, it's also been a really successful partnership with Ovarian Cancer Australia, who are one of our leading organisations for supporting women with ovarian cancer over here. And that original partnership, the intention was actually to help secure funding, of which it was a successful partnership in that respect. And in fact, we were able to secure funding from the Australian federal government for this work. But OCA have actually been incredibly valuable contributors to the study in a more practical sense as well, including, in really engaging their network of ovarian cancer patients and their families to promote and support the study.
And there are 3 arms to this study, aren't there? Do you want to explain a little bit more about that, how it works?
Yeah, sure. So as Rachel kind of touched on, the idea of Traceback is to go back and to find women who were diagnosed with ovarian cancer between the years of 2000 and 2016. That's what we decided to focus on, who, if they'd been diagnosed today, would have been eligible for genetic testing, but at the time missed that opportunity. Perhaps they didn't have any of the hallmark flags that clinicians looked for at the time, like a strong family history of ovarian cancer, and didn't actually receive testing. So we approached that to try and find these women. We recognized that most of these women, unfortunately, are probably now deceased, and we used 3 different approaches to try and find them. The first was working with existing ovarian cancer research studies that exist here in Australia, including the Australian Ovarian Cancer Study and OPAL, which is another big lifestyle and prognosis study that runs here. And this was quite easy to do. Women who were diagnosed with cancer were consented into these studies and donated blood samples and tissue samples and gave their consent for these samples to be used in future ovarian cancer research. So that was all very straightforward and a great resource for us to use. Our second approach was probably the most ethically challenging approach, and that was working with specialist gynae-oncology clinics from around Australia and getting them to audit their medical records to find these women who they'd seen during that time period and who, as far as they were aware, had no clinical genetic testing offered to them and who were now deceased. And the idea was that we were going to go back to pathology laboratories and access archival formalin-fixed paraffin-embedded tissue from these patients to then, use for this genetic testing. And this was going to be done under an ethically approved waiver of consent. And the third arm was actually one that we worked very closely with Ovarian Cancer Australia on, and that was a self-referral arm. This is when we actually advertised the study and reached out to ovarian cancer, patients and their families to bring themselves forward to consent into the study on behalf of their deceased relatives. And through that arm, we were actually also able to accept self-referrals to the study from women living with ovarian cancer now who haven't received genetic testing yet. But rather than doing the research genetic testing on those patients, we funneled them and helped them access actual clinical genetic testing that was going on that they were eligible for here in Australia. And again, with that arm, we were looking at accessing archival formalin-fixed paraffin-embedded tissue for these deceased patients.
So you were looking for signs that women had a genetic germline mutation rather than a somatic mutation.
That's right. So we used the archival tumour, the tissue samples. I guess we had them assessed by a pathologist. And what we were trying to do is, even though most of those tissue samples were cancer samples from the women's ovarian cancer surgery in most cases, we were using a pathologist to help us isolate areas of that tissue that was still normal or considered normal cells and use that DNA from those specific areas of the tissue to be more confident that the testing that we were doing was identifying germline mutations rather than somatic mutations.
So then the challenge begins, and Rachel, I'm going to turn to you on this one. So do you only try and contact families where a positive genetic mutation comes up?
Yes and no. So I guess there was a couple of parts to that because there's 3 components of the project. So the participants for whom we did give the information back, whether we found something or not, were for the self-referrals. So those particular individuals had come to us, they knew the testing was occurring and they were expecting a result. So we did pass that information back to them. We told them that either a pathogenic variant had been detected or that nothing clinically significant had been detected in that testing. So that was one group. For the other groups, only information if it was deemed to be clinically significant, as in a C4 or C5 pathogenic variant, and actionable, was then passed on. And that there were slightly different ways that information was passed on depending on which group that information went to.
And the most challenging group to reach, I understand, were those families of women who had passed away some time ago.
Yeah, absolutely. So that was one of our concerns when we started the project, and was thinking about how to approach the deceased cohort. And it was raised that perhaps we should try and reach the family members of those individuals first, seek consent, and then test. We were concerned, though, that it'd actually be quite difficult to do that because, as you are all aware, medical records are notoriously poor, and particularly the records kept many years ago. Some of these women were diagnosed more than 20 years ago. So finding a person that's appropriate and a familial contact for them, we thought would be a challenge. And that was one of the reasons behind the consent waiver. And actually, when we put that into practice, that's absolutely what we found. So finding a contact person was challenging in itself. Ensuring that the contact details for that person were correct was the next challenge. And once we were confident that all the information was correct, they received a notification letter. And then came the final challenge, which was actually to try and be able to engage those participants in a meaningful discussion, keeping in mind that the family member of that individual might have died 20 years ago, and that this phone call was coming for some quite out of the blue, even though they had received a letter. The letter was worded in a way that may not necessarily have been clear that there was genetic information available, and it was deliberately worded in a way to really ensure that people had the opportunity to decline knowing about that genetic information. So the challenge came with that was that sometimes that conversation was challenging.
So the initial letter didn't say, we have found a BRCA positive mutation. It just said there is something that we would like to talk to you about, sort of thing.
Yeah, that's correct. For those clinic cases where we were entirely aware that the testing had been done with the consent waiver, we were very aware of the ethics around that. It was worded in a way that did not state that genetic information was available, really rather sought their interest in knowing more about health information.
And then that follow-up call, you found it was really important that somebody who was very experienced or respected made that call as well.
Yeah, I think in such a short, that phone contact is completely out of the blue. And what our team found was that you had a really short amount of time to engage that person's interest. And so if the phone call could come from someone that was actually trusted and known to the family, already there is, through that rapport that's been built, you're much more likely to engage in a meaningful discussion and really be confident that that person's able to get enough information to make an informed decision.
And what has been the response of these families to this information?
Yeah, on a positive note, it's actually been quite good. So I can say so far, no one's been unhappy with the contact. And even when the contact comes out of the blue and the testing was done with the consent waiver, most people decided to go on to have further information from the familial cancer clinic, so to accept a FCC referral. So overall, we have been quite happy with the outcome so far.
And the idea is that then they go on and perhaps get tested themselves and then seek pathways that may reduce their risk.
Yeah, that's correct.
Kath, how many women are we talking about in this, and what stage is the project at?
Yeah, so the Australian Traceback study, we've always considered it a little bit of a pilot study for the Traceback, the idea of Traceback. As Rachel mentioned, there was the international workshop that happened a couple of years ago. So we do know that there are other countries around the world who are also looking at starting their own Traceback studies. We were the first cab off the rank. We managed to get this up and running quite quickly and quite smoothly. We were aiming to do 1,500 ovarian cancer patients, genetic testing on 1,500 patients. Look, COVID has reared its head, as it has with so many other things. We're a little bit delayed, but we are closing in on that number now and should have all that genetic testing finished by the end of this year. But look, hopefully this is just the first, the pilot for Australia, and, just the first one of these projects to get up and running around the world.
And what would you say, Kath, are the most important factors to get right in a project like this?
It's a really tricky project, the way that we've approached this with the 3 different arms. There's definitely pros and cons to each of those different consenting sort of pathways to be able to identify and test these patients. And a lot of that is probably very specific to different countries and different groups, what resources they've got available, what their overarching human research ethics sort of guidelines and things like that might allow. It's probably the biggest strength with Traceback is how collaborative it has been and how many specialists we've had really getting involved in the study from across the spectrum. Obviously people who are very experienced in the lab-based side of things, but then also in the genetics and the genetic counselling side of things, the clinical side of things, and of course OCA as well with the consumer engagement, which has been really, really important. And also, of course, funding is important for all of these things. And so we have to do, acknowledge and thank the Australian government for stepping up and funding this project.
And Rachel, turning to you, what do you hope for the project in the future?
So once we reach our 1,500, I think it would be fantastic to be able to extend the project to the other clinics around Australia. So we've had many groups come forward to us and expressed interest, with many clinicians know of the families that are out there and untested. So I think it is a real shame if we're not able to provide this service to them. So I think if we could concentrate on the major gynae centres that we haven't been able to test so far, that would be extremely important step forward for the project. And also what we found that the self-referring families are really a highly engaged group. So if we could potentially help them be able to access funded testing, potentially even through the government, I think that would be a wonderful thing to achieve.
At the moment, is genetic testing, you have to pay for it yourself in Australia unless it's part of a study?
So women that have cancer, or a person that has cancer, can have testing that's funded through the government. But unfortunately, if a person is unaffected, even if that is a first-degree relative of a woman with ovarian cancer, they don't get genetic testing that's funded. So that would have to be out of pocket, or otherwise through a study.
Okay. Well, Dr. Rachel Delahunty, Dr. Catherine Alsop, I wish you all the best for a continuation of this really important project. And thank you for sharing your insights with us today.
Our pleasure. Thank you for having us.